So I'm going to explain the process of aerobic cellular respiration to you, which is a process that takes place in the cells of living organisms, at least those that use oxygen in this process, to produce ATP by breaking down glucose. So the process takes oxygen and it takes glucose, and through a series of chemical reactions inside the cell, partly in the mitochondria and partly in the cytosol, we're able to produce carbon dioxide as a byproduct, water as a byproduct, and 36 to 38 ATP, which the cell will use to drive cellular processes. So what we're going to look at today is this bit around the arrow here, the chemical reactions that take place that enable us to break the glucose down in the presence of the oxygen to produce our outputs. So as I said, it takes place partly in the mitochondria. So I've drawn a mitochondria here on the diagram and labelled our main parts. And the main parts of the mitochondria we want to be familiar with are the outer membrane, which is the phospholipid bilayer surrounding the mitochondria, the inner membrane, which is a highly folded phospholipid bilayer inside the outer membrane, and those folds we sometimes refer to as cristae, and the folds give it a really high surface area. Between the inner membrane and the outer membrane, we have a space called the intermembrane space, and in the middle of the mitochondria you can see that area in the middle there is called the mitochondrial matrix. Assuming this mitochondria is within a cell, the fluid surrounding the cell would be the cytosol. So our aerobic cellular respiration is a three-step process. Step number 1 is glycolysis. Then we have step number 2, which is the Krebs cycle. And then we have step number 3, which is the electron transport chain. And they do go in that order. So let's have a look at what's occurring in glycolysis. So during glycolysis, we take glucose, and through a series of chemical reactions, the glucose is converted into 2 molecules of pyruvate. In breaking the glucose down into pyruvate, we release hydrogen ions and electrons, which we essentially want to use in the electron transport chain. So to pick up those hydrogen ions and electrons and transport them to the electron transport chain, we have our acceptor molecule coming in, which is the coenzyme NAD, and our acceptor molecule gets loaded with hydrogen ions and electrons, and then it leaves glycolysis in its loaded form, which is NADH, and that'll travel over to the electron transport chain. In breaking glucose down into pyruvate, we also release some energy, and that energy is used to phosphorylate 2 ATP, so to convert 2 ADP plus PI molecules into 2 molecules of ATP. So we would say glycolysis has a net yield of 2 ATP. And that's glycolysis done. So glucose is broken down into 2 pyruvate, NADH, and 2 ATP are produced. In step 2, we have the Krebs cycle. And the Krebs cycle essentially takes pyruvate and breaks that down into carbon dioxide. Now, in VCAR, you will talk about the Krebs cycle doing just that, taking pyruvate and converting it into carbon dioxide. In reality, though, there's a little step that comes before that, and there's a reaction we call the link reaction that combines or links glycolysis to the Krebs cycle. And this is what happens. So glucose itself cannot enter the mitochondria, and that's why we need to break that down into pyruvate. But pyruvate can enter the mitochondria and does. So pyruvate enters the mitochondria. I might show it just up at the top there. So this occurs in the matrix. And pyruvate does not directly enter the Krebs cycle, but first it gets converted into what we call acetyl coenzyme A, which is a coenzyme that takes the carbons from pyruvate and incorporates them into the Krebs cycle. So what the Krebs cycle does is it takes the carbons that are incorporated into it from acetyl-CoA, or in VCAR terms, it takes pyruvate, and through another series of chemical reactions, it is broken down to produce carbon dioxide. See if I can pick up that little carbon dioxide with my stylus. There we go. So the pyruvate has been broken down by the Krebs cycle into carbon dioxide. Once again, by breaking pyruvate down into carbon dioxide, we've released some hydrogen ions and electrons, and we want to transfer them to the electron transport chain. So once again, we have our coenzymes in their unloaded form coming in. Trying to pick up NAD, there we go. NAD coming in, and it picks up its hydrogen ions and electrons, and then obviously leaves in its loaded form of NADH. In the Krebs cycle, we also have another coenzyme that helps in the process, and it's FAD. So FAD also comes in. I might draw him over here. It picks up some hydrogen ions and electrons, and it leaves in its loaded form, which is FADH2. So pyruvate's been broken down into carbon dioxide, releasing hydrogen ions and electrons, which have been picked up by NAD and FAD, turning them into their loaded forms of NADH and FADH2. And both NADH and FADH2 will exit the Krebs cycle and deliver their hydrogen ions and electrons to the electron transport chain in the inner membrane. Also, by breaking down the pyruvate into carbon dioxide, we've once again released some energy that is used to produce ATP, and once again we produce 2 ATP. So we could draw 2 ADP plus PI coming in, and then we have a net yield of 2 ATP. So of our 36 to 38 ATP in total, so far we've produced 4. So obviously if you ever got a question saying which stage of cellular respiration yields the most ATP, you're about to see how it's the third stage, the electron transport chain. So let's move to stage 3, the electron transport chain. As I said before, the electron transport chain takes place here within all of the inner membranes, within those tightly folded inner membranes. However, it's really hard for me to draw it there, so we're going to imagine that out here is a magnified version of the electron transport chain. Let me get rid of these. Don't know why they're there. They're pretty much doing nothing. So I'm going to represent the phospholipid bilayer of the inner membrane just as a nice little diagram like that. And in our electron transport chain, essentially we have a series of proteins that make up the electron transport chain, and at the bottom of our electron transport chain we have a particularly important protein which is an enzyme, and that enzyme is called ATP synthase. ATP synthase. And ATP synthase is an enzyme that can take ADP plus PI and convert it into ATP. And in the electron transport chain, ATP synthase produces 32 to 34 ATP. The thing with ATP synthase is it's not like a normal enzyme. For it to be activated to catalyse that chemical reaction to produce the ATP, it actually needs to be activated by hydrogen ions flowing through it. So it's like an enzyme and a channel protein all in one, and it needs hydrogen ions to actually flow through it, and by the hydrogen ions flowing through it, it's activated. And that process is called chemiosmosis, the process of the hydrogen ions flowing through it. So what we need to do in the electron transport chain is set up a situation that ensures a steady flow of hydrogen ions will continually flow through ATP synthase so that we keep producing ATP. And if you can appreciate, that's why I had these little things here, because this picture is a representation of the inner membrane, the inside of the picture is the matrix, and the outside is representing the intermembrane space. So we need to set up a situation where hydrogen ions flow from the intermembrane space through ATP synthase back into the matrix. And this is how we do it. So as the name suggests, the electron transport chain is a chain that transports electrons. We need to provide it with electrons. And the way we provide it with electrons, if you remember, is in glycolysis we produced NADH, so it would travel over to the electron transport chain. And then in the Krebs cycle we produced FADH2, so it also travels over to the electron transport chain. And also in the Krebs cycle we produced more NADH, and it also travels to the electron transport chain. So we're supplying the electron transport chain with the hydrogen ions and electrons that were produced in glycolysis and the Krebs cycle. You might also remember that as an input over here we've got oxygen, which we haven't accounted for yet. Oxygen is an input of the electron transport chain. If you ever get a question that says which stage of glycolysis is oxygen an input, your answer is the electron transport chain. So oxygen actually sits at the bottom of the electron transport chain. So here's what happens. NADH and FADH2, they release the electrons they are carrying into the electron transport chain, and those electrons travel down the electron transport chain and they eventually combine with oxygen. And the thing dragging or pulling the electrons down the chain is the fact that oxygen is electronegative, which means it has a strong affinity for electrons and the electrons are attracted to it. And the proteins making up the electron transport chain are the things that are transporting the electrons down and also breaking its fall, so it's a controlled release of energy, because that's exactly what's happening. As the electrons fall down the chain, some energy is released and we can use that energy. So NADH and FADH2 have dropped off their electrons. The electrons have entered the electron transport chain. You should also remember that NADH and FADH2 were carrying hydrogen ions, and they dropped the hydrogen ions off in the mitochondrial matrix just beside the inner membrane. So as I just said before, by the electrons falling down the chain, we release some energy. That energy is actually used to pump hydrogen ions and transport them across into the intermembrane space. So we would get a large accumulation of hydrogen ions in the intermembrane space. And the way the hydrogen ions move into the intermembrane space is via those proteins that are making up the electron transport chain. They're able to transport hydrogen ions from the matrix across into the intermembrane space. However, they're only able to transport in one direction, from matrix to intermembrane space. So what we end up with is we end up with a high concentration of hydrogen in the intermembrane space and a lower concentration of hydrogen from the matrix, because we keep pumping it out of the matrix into the intermembrane space. Hydrogen, like other molecules, will naturally want to go from an area of high concentration to low concentration via the process of diffusion. So the hydrogen ions will want to diffuse back across the membrane in the aim of reaching equilibrium. And they have a passage to get back across the membrane, but they only have one passage. It's not the proteins they came through, because they're one-way channels. It's ATP synthase, because it is the only channel that will enable the hydrogen ions to move back from the intermembrane space into the matrix. So the hydrogen ions are all forced to travel back through ATP synthase. And that is a good thing, because we started this big long story by saying ATP synthase makes ATP, but it must have hydrogen ions flowing through it. So the hydrogen ions pass through, fantastic, ATP synthase can now make ATP. That's great. However, as the hydrogen ions enter back into the matrix, well, they're going to start accumulating. And if we don't get rid of them, they'll accumulate to a level where they can eventually become equal with the concentration in the intermembrane space, and then diffusion won't work and ATP synthase won't work. So what we say we do is we maintain a hydrogen ion concentration gradient. We maintain a high concentration in the intermembrane space and a low concentration in the mitochondrial matrix. And how we're able to do that is, as the hydrogens move back through ATP synthase into the matrix, they actually fuse with the oxygen. So 2 hydrogens at a time fuse with 1 oxygen. And if 2 hydrogens are bonded with 1 oxygen, well, what we produce is water. And that's why water is an output of cellular respiration. So what we say is the oxygen mops up the hydrogen ions. By mopping them up, it means the oxygen continues to get them out of the matrix to maintain the concentration gradient. And it gets them out of the matrix by bonding with them to form water, which is a byproduct that can leave the mitochondria. And that's it. That's the process. So electron transport chain is definitely pretty confusing. If we were to summarise it really simply, or maybe I'll summarise the 3 of them really simply. Glycolysis, glucose is converted into pyruvate, into 2-pyruvate. In doing that, NADH and 2-ATP are produced. Krebs cycle, pyruvate is broken down into carbon dioxide. In doing that, NADH, FADH2 and 2-ATP are produced. In the electron transport chain, NADH and FADH2 deliver hydrogen ions and electrons to the electron transport chain. electrons travel down the electron transport chain and fuse with oxygen. Hydrogen ions are pumped into the intermembrane space and pass back through ATP synthase to generate ATP. Hydrogens fuse with oxygen to form water to maintain a concentration gradient to ensure that ATP synthase continues to produce ATP. So that's the process. Pretty complex, but you'll break it down in class with your teachers as well. Hopefully this is just another little thing that helps you to understand.